Okay, well, thank you very much, the organizers, for inviting me to present to you guys today. So I'm a comparative immunologist. We're not just interested in birds, we're interested in immunology across the species and try and use comparisons to actually understand what the fundamental aspects of immunity are. So today, I am going to deal very much with immunity, and perhaps the title I have on my talk gives you a basis for what I was asked to do. And actually, I was asked to consider whether we should use nutritional interventions that are either pro-inflammatory or anti-inflammatory. And really, my bottom line on this is actually we should be looking for balance. And it's been mentioned one or two times before, balance in terms of microbiota and things like that. Immunity is just the same. There is no one easy way of describing what we should be doing. So what I hope to do over the next 40 minutes or so is to literally take you on a journey through different types of and areas of immunology, how it relates to microbiota, and I'm going to give you a little example of what happens in terms of microbiome immune cell interactions, both from perhaps the perspective of the immune system and also from the microbiota. So I hope you find it interesting. So, the first thing that I did was I surveyed a whole bunch of the literature in terms of what it was that we were trying to answer the question that I was set. And really, the question very much is, what are we steering? What on earth is it that we're trying to steer in terms of bringing birds from the very young age to birds that have finished their production cycle? And the bottom line is, it's an absolutely moving target, because what we're dealing with is dealing with birds that are really changing immensely over that period of time. And if we think about all the possible influences on gut health during that time, and these are just some of them, really one of the central players is actually the microbiota. And much of the time when we change the food or change the or add components to our food, what we end up doing is modifying the microbiota. The microbiota interacts with host genetics, physiology, immunity, interacts with other aspects of the food, and of course interacts with pathogens at the same time. So what we're dealing with is actually a really complex scenario, and we mustn't forget that. It's very, very easy to measure one or two things and think that we've got the answer. I'm going to end up encouraging people to measure things much more broadly in terms of trying to understand the interactions, and hopefully that will allow us to understand the mechanisms that are operating. So one of the other classic scenarios, if we were to think of the evolution of the interaction between the gut immune system and the microbiome and the food and all those sorts of events, is that we must recognize that what we're doing in the poultry industry is we have changed the environment and the bird immensely. They're completely fine-tuned for production, not necessarily for gut health. And now what we're trying to do is to reintroduce some sort of natural scenario where if you look in evolutionary time, birds and their microbes, or all animals and their microbes, have actually come to a position of balance. So what have we done? We've changed the genetics. We massively changed the numbers that are there. The production systems, the speed at which these birds are growing is unbelievable, and certainly not a situation that would have been present at the times when the whole interaction was evolving. One of the other big effects that we do is we segregate the chicks away from the maternal environment. We don't just segregate them physically by taking the eggs away and hatching them. What we do is we disinfect the eggs, we disinfect the environment, we try and keep them as clean as possible. This is completely unnatural. And it's hardly surprising that we end up with issues with gut health when we're trying to do so many things. And I think what we've got to try and do is to bring the controlled aspects of creating a natural environment as we can, and that will start helping us understand, or help us control various gut problems. So I'm going to deal with a whole raft of different things today, and I just thought I'd sort of introduce some of the scenarios, some of my pains of reading was that I tried to look at all sorts of different foods and additives and what did we know about the mechanisms. And one of the things that I'm most interested in is not just describing what happens, but actually understanding mechanistically what's happening. And I really quite struggled in the sense that a lot of different tests are done, but they're rarely sort of comparable between different types of studies. And we've got all sorts of different ways that things can be modified. And at the end of the day, one of the conclusions that I really came to was that the central component, as it was in that previous slide, is that it's the microbiome that seems to be influenced really quite dramatically during that early stage of life. And that can alter the gut immune system immensely, which then in return alters the microbiome. And then what you end up with is a cascade of events, both the host and the microbiome. We shouldn't be thinking of them as separate entities. We should be thinking of them as a single holobiont and actually a changing circumstance over time. One example I've got here from some of Ivan Reichlich's work, some of his published work, is actually looking at some of the changes in the microbiome over time. And I'm not going to go into any details here, except to say, in the very, very young animals, this is changing incredibly rapidly. These happen to be layers out on a commercial farm that he was looking at the microbiota over a long period of time. So there's a big change in the first few days of life. Those changes continue for many weeks. And actually, in the commercial layers, you're out there at about 20, 25 weeks before you get what we might consider a stable circumstance. But actually, the whole thing is stable because it's quite reproducible. Those changes are actually normal. But we have to be thinking that it's during those very rapid changes that a small influence can lead to really, really big downstream effects. I was asked to talk about immunity. Now, I'm acutely aware that there's a whole bunch of people in the audience who will now basically be going, I'm going to switch off. This immunity stuff is too complex. I'll try and keep some fairly simple language trying to explain the concepts that we're dealing with. The gut is the most complex and the largest immune organ in the body of any animal species. It's bigger than if you put all the other immune compartments together. It's got loads and loads of different populations of cells, and these populations of cells do different things. But the other important thing is these populations of cells interact with each other and are interacting with the external environment that is the lumen of the gut. There's cells like macrophages. I'm sure most people have heard of those. They're like the dustbin men, the dustbin lorry of the immune system. You've got other cells like dendritic cells, which drive this group of cells called T cells into dividing, and that's going to become quite important as we move through the talk. We've got B cells. They're the cells that make antibody, and we've got the natural killer cells. And many of those components you'd find all over the body, and they link between other parts of the body and the gut. But when you look in the gut, you've got overrepresentation of some components, and I've just kept this quite simple. You've got lots of B cells that are producing IgA, the secretory antibody. You've got rather special dendritic cells, and I'm going to deal with that in a general situation in a few minutes. And in birds in general, we've got a high number of these rather unusual T cells called gamma delta T cells. Now, to be honest, I'm fascinated by them because they break all the rules. We don't know much about their biology in any animal system, and they're most studied in mice, for example. But mice don't have very many circulating gamma delta T cells, so they're most studied in a system that hasn't got very many. Birds and many other animals have very, very large numbers of gamma delta T cells. This is where we're going to work out how they function. The other point that I wanted to make is that the epithelial cells themselves are part of the immune system. They are cells that are able to produce effector molecules like defensins, and they can also be really intrinsically involved in activating and calling in cells of the immune system into the gut. So they're often neglected, and they shouldn't be neglected in terms of the interaction between the microbiota, because of course they are indeed the interface between the internal and external world. Now this is a really sort of simple slide that I'm just going to point out for those that are sort of not immunologists, the sort of main events that happen. So really what we've got is we have detection events, and there's a bunch of receptors called pattern recognition receptors, and for some of you, These include things like the toll-like receptors. I'm not really going to talk about those today. But they're about recognizing what's good and what's bad, essentially. And they initiate the whole response. Initially, the innate immune response, that then moves on to the adaptive immune response. That takes time to develop. And the outcome, hopefully, in terms of pathogens, might be pathogen removal or killing, or control of a pathogen at least. Problem is quite often the immune system is responsible for nearly all the pathologies that we actually see associated with pathogens. Relatively small amounts of it is directly driven by the pathogen. The vast majority is driven by the immune system. So if we have a sort of gut epithelia here, this is one of the innate cells of the dendritic cell. Here's a macrophage. These will engage with any microbes or pathogens, particularly if they try and cross the epithelia and get into the deeper tissues. The one difference is that the macrophage will try and kill in situ, whereas the dendritic cell is a bit of a scaredy cat. The moment its TLRs are tickled, it runs away. And it runs away and goes and talks to T cells, makes them divide. They then go back, have a chat with B cells, make antibody like IgA, or superactivate the macrophage. That is the basic sort of mechanisms that operate in immunity, and we're going to deal with some of those events in turn. So let's start off with the dendritic cells. Now, I said there's quite a lot of immune cells in the gut. These particular images happen to be of a mouse, where all of the dendritic cells have been lit up with GFP, okay? So all of the green in this gut is actually dendritic cells. And that shows you the intensity of the network of these first-level immune surveillance cells as they're sitting there in the villi. There's a huge number of these cells waiting for action to happen. Some of them don't even wait for the action to happen. They stick their dendrites across the epithelial barrier and wave them around out there where all those microbes are out in the lumen of the gut. Functionally, what happens is that there are two real scenarios in the gut, one of which is a steady state. So they're always entering and leaving the intestine at a pretty steady state. And if they do that, then what happens is they have a prolonged period in the intestine, and the dendritic cells that leave here are generally involved in regulation and induction of oral tolerance. If something goes wrong in the intestine, then the dendritic cells spend less time essentially in that environment, and when they exit, they're much more likely to be inflammatory and drive classical inflammatory immune responses. Now, those sort of effects are generic effects that occur in the gut, but there is one other thing, and I was asked to talk about diet in terms of immunity that I find really quite neat. And this is literally the story of the carrot. So what happens is these dendritic cells that are conditioned out there in the gut are exposed to the environment in the gut, or associated with the gut. And that includes a molecule called vitamin A that might be quite familiar to us. And the vitamin A turns out to be really important in the immune system. Because what happens is those dendritic cells out in the gut pick up vitamin A and also some metabolic derivatives of vitamin A called retinoic acid. And when they run off, and this would be a mammal, into a lymph node, but in a bird we actually don't know where they're running off to. When they run off, they then go and talk to naive T cells. The moment a naive T cell recognizes an antigen or molecule on the surface of those dendritic cells and starts to divide, it actually puts on a bunch of receptors on its surface. One of those receptors is called the retinoic acid receptor. And if that cell, in its first point of division, experiences retinoic acid derived from vitamin A, then that T cell will then change its surface and put onto its surface homing receptors that will send it back to the gut. So here the dendritic cell that's exited the gut is carrying with it an address, and it hands that address across to the T cell and makes the T cell go back to the gut. And that is derived, but really based upon the environment, and in this case, a food-associated molecule, vitamin A. I'm now going to move on to some of the T cells. So these are some of the T cells I was asked to consider inflammatory versus regulatory signals. Now, these T cells, many of them are what we would call inflammatory or will actively engage and try and kill pathogens. But there are some subsets of T cells that go, calm down, guys, they're the regulatory T cells. And actually, if we look at normal components of the microbiota, we already know that they drive preferentially different types of cells. So the SFBs, the segmentofilamentous bacteria, really strongly drive a pro-inflammatory cell type. Some of the strains of Clostridia really strongly drive regulation. And others really strongly drive both under different circumstances. And it's probably about the environment they find themselves in as bugs as well. So what we've got, the most important thing here is actually balance between pro-inflammatory and regulatory. If you've got too much inflammation, you'll get pathology, you'll lose those villi. If you've got too much regulation, then you won't be able to actively respond to any incoming pathogens. What I'm going to do now is to move on to some of our data and actually talk about some of the processes that are involved. And the first thing I'm going to do is I'm going to take things right to the extreme. So what happens if we massively restrict the microbiota and look at germ-free or notobiotic birds and then ask questions about how is the immune system developing, what are the influences of the microbiota in terms of this? What we know is the gut structure is affected, particularly the layer that lies just underneath the epithelia called the lamina propria. It's full of immune cells. That is massively restricted in terms of the number of immune cells that are entering the tissues. The numbers of classical T cells and B cells are actually reduced, and I'm going to show you some data on that. And then I'm going to move on about the diversity of the receptors that are actually being induced in these animals. So, if we look at the macrophage as an innate cell type in germ-free individuals versus conventional versus individuals that have just received a single monocolonization with E. coli or 4 different strains of bacteria, there ain't much difference at all. They've all got macrophages in their intestine. They're driven there in a microbiota-independent fashion. In contrast, if you look at B cells, I think you'll see a difference. So this is immunohistochemistry for B cells. So this is a conventional animal, lots of B cells in the intestine. This is a germ-free, well, they're few and far between in the intestine. And if you put a single E. coli in, you get a few spots, a few more spots if you count them. But if you put 4 bugs in, then you start to see reconstitution of the phenotype. But this is just the number of B cells that are there. The question is what are they recognizing? So in this situation, these are using an antigen that's based upon the E. coli, which was the single colonizer. Germ-free animals don't have any IgA against E. coli. The monocolonized with E. coli have strong responses against E. coli. If you put that E. coli in the context of 3 other bugs, those responses against E. coli are still detectable, but they're actually reduced in terms of overall magnitude. And if you look at a conventional flora, they're really very, very low indeed. If we take another bacteria, this is Epithelium, these are the same groups of birds, so we can tell that this antibody is absolutely specific because the mono E. coli exposed birds are not reacting to this Enterococcus. The tetrabirds are reacting to them, but are actually at a much lower level than we see against E. coli, so there are differences between the different microbiota components, and the conventional birds are really quite variable in terms of their recognition. So we've got recognition. How can we follow some of the events that are going on? And one of the things that we've been doing quite a lot of that's a little bit unusual is actually trying to understand the repertoire of these T cells and B cells that actively respond and have the effector function. And we use 2 different methods. We use one method which essentially gives you a profile. So the normal profile would look like this, lots of different peaks in terms of the receptor profile. This would represent a normal population of cells where every cell has got a different receptor on. So this is what happens with adaptive immunity. in this room, I haven't counted up how many people are here, but each and every one of you is carrying about 100 million different adaptive immune receptors, so we don't like sequencing them all. But that's what the population looks like. If you get a pathogen come in or an antigenic stimulus, the most important thing that happens to these small, rather insignificant cells, they really are not very flashy in terms of looking at their structure. The most important thing that happens is that they divide. So I use the analogy that the macrophage was the sort of dustbin man of the immune system, clearing up all the mess. And being in Rome, I think I should use a slightly different analogy for the lymphocyte. This is essentially the Ferrari of the immune system. If you want a car to go really, really fast, you don't put all the extra bits on. You take it all off. And a lymphocyte, which is the key cell in adaptive immunity, is basically stripped bare of anything that it doesn't need. So the one thing it can do is replicate really fast. And that's absolutely key in terms of adaptive immunity. So we can follow those receptor changes, and they tell us about the populations of cells. So in a normal, unselected population, we see a diverse repertoire of cells. In a selected population where one clone has grown out, we see a much more focused population of cells. And if we look in different situations and look at the repertoire, in this case of alpha-beta T cells, what we can see is if we look in conventional animals at the repertoire of alpha-beta T cells, big normal distribution in the spleen, but wherever we look in the gut, what we get are actually biases in that repertoire. And that's because there are local T cell clones that have grown up that are bigger than they were in the fundamental initial repertoire. So we see that complexity in terms of the repertoire in the gut and in different regions. Sometimes you've got shared clones, sometimes you've got distinct clones. But if you look in a germ-free, then basically what you get is a normal distributed repertoire all over the place. You've got no selection that's happening. What's interesting is when you culture, in this case a monoflora with bacillus, is what we ended up seeing was a single clone of cells that was outgrowing in all portions of the gut. And we were able to show that this was actually actively responding to the bacillus. So that's what happens if you have different types of gut microbiota and T cells in terms of alpha-beta T cells. And I'm going to move on to this other group of T cells, which is the gamma-delta T cells. And these are, as I said, are rather odd. They don't follow the same rules. There's lots of questions about whether they're part of the innate or adaptive immune response. But what I think they are is probably part of both. And I'm going to show you about microbiota-dependent and independent biology of these cells. I think these cells are quite fascinating and underexploited in terms of being able to improve gut health. So if you look at the simple gamma-delta repertoire in the spleen, there are 3 large populations of gamma-delta T cells in the chicken. What you see in colonized normal individuals and in germ-free individuals is pretty diverse repertoires. In all of the 3 different V-gamma populations. And we got really sort of trying to worry about subtle differences between the two. And then we looked in the intestine, and in colonized individuals, you see some clonal biases, but a pretty diverse population. But when you go into germ-free individuals, in V-gamma 1, it's polyclonal. In V-gamma 3, it's polyclonal. We got a little bit of a surprise because in one of those, in the V-gamma 2s, we basically saw a single peak on this particular graph, and when we sequence those T cell receptors, they're all exactly the same clone. So there's this large monoreceptor clone that's in the gut of germ-frees at very, very high levels. When we look at a lot of individuals in both the small intestine and in the large intestine, what we see is this large clone is present in the small intestine of all of our germ-frees and in the large intestine of all of our germ-frees, and also present in the large intestine of actually our colonized individuals at really high frequencies. I said that when we sequenced it, this is essentially the same receptor everywhere. This is the receptor sequence in the germ-free ileum and the germ-free colon, and actually in conventional colon we were also able to see this particular canonically rearranged sequence. By canonical rearrangement, it's essentially taking the segments that make up the T cell receptor and sticking them together in the germline configuration. Now, there are other canonical gamma delta T cells that have been studied in the mouse, and these gamma delta T cells include the V gamma 5s and the V gamma 6s, which are the very, very first waves of gamma delta T cells ever made in the mouse. And the V gamma 5s all go to the skin, and the V gamma 6s all go to the uterus and to the tongue. The third wave of gamma delta T cells are V gamma 7 in the mouse. They're not canonically rearranged, but quite restricted, and they all go to the gut. So you've got these waves of gamma delta T cells that are protecting the epithelia. And we wanted to ask about the biology of this rather unusual, canonically rearranged gamma delta T cell in the chicken. And what we know is that gamma delta T cells are found in the earliest waves of thymocyte development, but unlike what we see in the mouse, what we could detect was all of the different TCR gamma chains being rearranged at the same time. But when we looked at the ontogeny of this particular V gamma 2 population of cells during embryonic development and early period post-hatch, what we see this is the peak that we're interested in, is that at 19 days of embryonic development, you've got this dominance of that clone, which isn't so strong at 16 days of embryonic development. These gamma delta T cells are beginning to exit the thymus at about 15 days of embryonic development, and then it maintains its dominance in this early period post-hatch. Looking at it at a slightly more fine scale, so if we look at 15 days, diverse repertoire, 16 days, diverse repertoire, 17 days, diverse repertoire, 18 days, diverse repertoire, bang. At 19 days, it's all changed. And now we have this very focused repertoire. There's something rather odd going on here. This is just before hatch. This is a gamma delta T cell, and what we know about gamma delta T cells is they interact a lot with epithelial cells that is homing to the intestine just before hatch. So what do we think is happening? This gamma delta T cell, just like the other epithelial gamma delta T cells we know of in the mouse, appears very early in the embryonic thymus. But it seems to migrate out of the thymus, and we can find it if we sequence at very deep sequencing in other places like the spleen. We can find it in the gut at very low frequency. But what happens between E18 and E19 is essentially all of these cells appear to migrate to the gut. And we think there's something changing in the intestine, and it's calling in these cells. And then these cells are actually involved in some of the late-stage embryonic development changes to the intestinal epithelia to prepare the bird for that horrible, exhausting environment of hatching out into the microbiologically rich world. Once the microbiota comes in, these microbes actually then drive the diversification of all the other gamma delta T cells and the V gamma 2s to form a diverse repertoire. And that's what we think that these cells are doing. Now I'm going to just change tack for the next few minutes and deal with microbiota immune interactions. And what I'm going to try and convince you is that the immune system is about stabilizing the complexity of the microbiota in all animals. I don't think this is a very special circumstance in birds. And what I'm going to try and sort of describe to you is based upon a conversation I had with some theoretical ecologists quite a long time ago. They can be very taxing conversations at times. But one chap just turned around to me and said, this microbiome stuff, it's too complex. It can't be stable. It doesn't fit with the theory. Well, I replied to him saying, well, unfortunately, we know the microbiome can be stable, so maybe the theory is not quite accurate. But that's like a red rag to a bull in terms of some of the opinions that we have to deal with. And then I wondered, what would happen if I could create lots and lots and lots of slightly distinct niches in the gut? And his response was, oh, that'd be okay. So the analogy I'm going to use over the next sort of few minutes when I'm going to just deal with this particular area is It'd be like trying to understand finches from the Galapagos Islands. If somebody come back with a shipload of finches and gave them to you and said, oh, look at these, there's lots of different ones, isn't there? They would really make no sense. But what was so crucial to Darwin is that actually he knew where they came from. The finches were different on different islands. And I'm going to try and convince you that the gut microbiome and the immune influence of the gut microbiome is based upon island ecology. So, that's told you the outcome of it. So if we think of the microbiota, and we've heard a lot about it, so I haven't really got to introduce it too much. Lots of people examine the poop. A few people examine the intestine. This happens, sorry, this is prepared for a mammalian audience, but examine the intestine. And one of the big issues is wherever you examine, what scale are we examining it at? You take a poop, you've got an intestinal sort of structure. This poop has bounced off one wall onto the other wall, and got mixed up a bit. People assume that this is ultimately mixed population. It's representative of what's happening in the gut. That's not true. It's some sort of random representation of what might be happening in the gut. Clearly the bugs are there. And sampling is really, really important. The scale at which you're sampling and the type of samples are really important, and I hope to convince you of this. And this was driven a little bit by some theoretical work done in in collaboration with Kevin Foster by one of our shared students, Kirsty McLaughlin, who essentially looked at a bunch of mathematical models, and I'm not going to go into the detail, but I'll give you the bottom line, is these models were very, very unstable in terms of which microbes won and which microbes didn't. So there was a stochasticity in the model. And that is somewhat familiar in some ways to some of the things that we're looking at. But if you actually can provide some sort of spatial structure to an adhesion molecule that the bugs could bind to, then this spatial structuring allowed spatial structuring of the microbial communities. And then that became much, much more stable, something we know actually happens. So one of the things that requires is some sort of contemporary spatial structuring of a host-provided adhesive molecule. But we need massive diversity. So we've got to explore what it is that adhesive molecule might be. And our strategy, and this sort of holds true in all sorts of different animals, our strategy was slightly crazy, and I don't think Kirsty's ever forgiven me for this, was to take a piece of intestine and chop it up. And chop it up into lots of tiny bits. So we have all the islands, carrying on with the analogy of the Galapagos. We have all the islands. So we chop it up into lots of different bits, and then what we're going to do is we're going to analyze the microbiome and the immune repertoire in every piece. So typically the size of the pieces have been 1 by 1 millimeter squared or 2 by 2 millimeter squared. We get quite similar results at those two scales. Yeah. So their first prediction from the model was we needed uneven distribution across microbes over small scales. Here's the grid, the intensity of the color. These are different microbes, microbe A, microbe B, microbe C. Doesn't matter what their identity is. The bottom line is, if we look across the grid, we see different patterns of different microbes. Their proportional diversity is very different across the grid. The second thing, we needed to get some form of spatial structure in the microbial communities, and we used a method which allows us to look at the similarity or dissimilarity between any two chunks on the entire grid. So we compare this particular chunk with every other piece on the grid, and then we ask the question, is the comparator chunk close to the original one or far away? And what we've done here is we've simply plotted the distance versus the dissimilarity between the different chunks. And as you move further away on the grid, then they get, on average, more dissimilar than neighboring regions. So we've got some sort of spatial structuring. What's quite cute is that you can then take this analysis and serially take one microbe or one sequence out of that microbiota analysis. And that's essentially bootstrap the scenario and then ask for what is the patterns of microbes that we see that when we remove them, we get an increased dissimilarity space association. Those are microbes that we tend to find everywhere, or a big decrease in the dissimilarity space relationship. And these are microbes that are very spatially structured within the very small segment, a whole centimeter of intestine. We then asked what happens in the same way to the adaptive immune system. IgA repertoire shows exactly the same pattern. As you move away in distance, we see all the fragments are different, but as you move away in distance, they're spatially structured. TCR-beta, the alpha-beta T cells, are doing exactly the same. We see spatial structure and association with the microbes. TCR-gamma, very different in every segment, lots and lots of difference, but they don't structure in terms of organization. So they're a very different relationship that's going on here. So really, I'm going to summarize this stuff before moving on. I'm going to skip the last section and move on to the end. And really what we've got here, I think, is immune-generated adhesion as a mechanism for spatial structuring of the microbiome. We've got an uneven distribution of the microbes across small scales. We've got spatial structuring of microbial communities across incredibly small scales. We've also got contemporary spatial structuring for host-provided adhesive molecules. IgA. IgA we all think of as being an anti-pathogen molecule. How about IgA actually being a community structuring molecule in its primary role rather than anti-pathogen? And we're trying to use this spatial structure to link community members, microbial community members, and IgA specificities. And then we want to ask questions, and this is where I appeal to you with all the different infection models, because I think that different infection models will disrupt this very delicate balance in the microbiome in different ways. Some by promoting high levels of IgA against the pathogen, others by doing different things. And what we're really interested in doing is actually exploring that. So how do we think this might work? We think that microbiota immune interactions are quite important. It's become quite topical in the literature. I think what we're putting on it is a spatial context that's a little bit different. So if we've got the mountains and the valleys that are represented by the villi, and you've got particular B cells producing antibody of particular specificities inside each villus. The antibody that's produced, the IgA that's produced, is going to be translocated primarily across the epithelia of that villus. It's not really going to diffuse over and get across into the neighboring villus. So what we think is you've got communities of B cells, whether it's 1 villus, 10 villi, or 50 villi, I don't know, but communities of B cells inhabiting space. And in a neighboring space, you've got another community of B cells. And a neighboring space, you've got another. And maybe it's repeated, depending upon how far away you're looking. These IgA molecules then essentially stick together. Fred and his mates here, George and his mates there, and Carl and his mates over here. And basically establishing founder communities that are differentially organized across space in the gut. If we disturb that with a pathogen, what happens is that this all gets into a big mix mash and these bugs start fighting with each other, and that's where we get unusual dysbiotic effects. But it recovers remarkably well. And the reason we think it recovers well is that actually these B cells are still there. They're just waiting for the epithelium to repair, and then they can reestablish their local communities. So that's the sort of research part. Now I'm going to skip through a couple of slides. I was going to talk a little bit about Campy. I thought this was too much, but let me just skip through these. And the paper hopefully will be out next week. And just really come to the last piece that I want to talk through with you. How do we assess the effect of manipulations? That was the task I was set. I've just picked a couple of little bits and pieces. Let us recognize it's a complex moving target. Massively complex, massively different. So what do I think that we should be doing? Yeah, we can all measure lots of parameters, and we should carry on doing so. Actually, we should measure as many different things as possible. We should apply multiple methods, but respect both their benefits in terms of interpretation and the difficulties in interpreting. We've got to be very, very careful in the way that anyone can apply a method. It's about interpreting it. And really, the bottom line is we've got to try and consider the biology. And I really am absolutely fanatical about the fact that we should be moving all the way backwards and forwards seamlessly between the field and experimental systems. All experiments are a form of model. All field situations are hyper-complex. But we've got to be moving backwards and forwards between these to try and collectively come up with some solutions. And that means using all of the tools that are available and generating some more, but really thinking very carefully about the function of the immune system. It's not about measuring stuff, it's about how it works. And that's an area that I think in the next few years we're going to see some big changes. Let me just thank you for listening, and the key players in my lab are listed here with their different areas. And this work would not have been possible without many of you that are in the audience and many people that you know that haven't made it here to this meeting. Those collaborations are absolutely key to us being able to do this work. Thank you very much.